Hi there, welcome to our IB Biology Revision Series, section A1.2 Nucleic Acids. So, DNA is the molecule that provides the long-term stored genetic information for organisms. DNA has bases, sequences. Of these bases, they are referred to as genetic messages, which are genes. The messages are codes for amino acids. Amino acids are the building blocks of proteins. Viruses are not living organisms. Some contain RNA in their genetic material, and some contain DNA. The subunits in both DNA and RNA are nucleotides. Nucleotides have sugar, deoxyribose in DNA, and ribose in RNA, a phosphate, and a base that contains nitrogen. So sugar to phosphate backbone of DNA and RNA. Nucleotides in both DNA and RNA bond together to produce long chains or polypeptides in order to form a chain of nucleotides. The pentose sugar of one nucleotide is covalently bonded to the phosphate group of the next nucleotide. The nitrogen base is DNA. So, one-ring bases, which are pyrimidines, are cytosine and thymine. The two-ring bases, which are purines, are adenine and guanine. So, the nitrogen bases, RNA. So, one-ring bases, pyrimidines, these are cytosine and then uracil replaces thymine. The two-ring bases, purines, adenine and guanine. So, the structure of RNA, the pentose sugar in RNA is referred to as ribose. They are single-stranded nucleotides which are linked as covalent sugar-phosphate bonds, and the example below shows a diagram of a condensation reaction of a sugar and a base. For an exam tip perspective, the bonding of a sugar to a phosphate is a condensation reaction, and we'll go through how to draw these when we do exam questions. So, the structure of DNA. DNA consists of two strands which are linked by hydrogen bonding. As outlined, base pairing exists between bases of DNA. Adenine pairs with thymine, cytosine pairs with guanine. These are referred to as complementary base pairing. Strands are antiparallel and run in opposite directions. These are wound together, which form a double helix. So, what is the difference between RNA and DNA? DNA, it contains a deoxyribose sugar, thymine as a base, the strand is two nucleotides, it's shaped into a double helix, acts as a permanent genetic code of a cell or organism. RNA, the sugar is pentose, the base is uracil, the strand is one nucleotide, and it has a variety of shapes depending on the type of RNA, which we'll go through in later sections. It does not contain a permanent genetic code except in an RNA virus. So, ribose has the molecular formula C5H10O5, whereas deoxyribose has a formula of C5H10O4. RNA has an alcohol or hydroxyl group. If you remove the oxygen, you are left with the hydrogen, and this is the difference between ribose and deoxyribose, and you can see this difference on the diagram below. Thank you very much for listening to this section on A1.2. We will be adding further revision videos into our IB Biology Revision series.